Okay, our next speaker is Jan Johansson from Artery Therapeutics, and we look forward to hearing what he has to say. Jan?
Very nice to be here. It's my third time. Thank you so much for inviting me. So ApoA-I is an ApoA-I HDL mimetic that reduces atherosclerosis with potential to prevent heart disease and stroke. I'm Jan Johansson, the CEO and president of the company. So the presentation follows Yogi Berra to some extent. You can learn a lot by watching, and if you do not know where you're going, you'll end up somewhere else. And that is associating with the context of the background of this particular biology, and also specifically the target. So we are a San Francisco Bay Area company focusing on translational medicine. John Bailicki here is here. He's from Berkeley, and he's the father of the technology. We're doing pharmacology mostly at Stanford, and it's all about treating atherosclerosis, which, as you know, is the biggest therapeutic group. And we have an IND-ready peptide, which is very promising. So just a little background, about 2 million people in the US get a myocardial infarct or stroke every year. It is the largest killer in the Western world, and it is very quickly becoming the largest killer in the non-Western world as well. And the cost in the US only is $450 billion per year, which is on par with BUD GDP of California. Statins and LDL lowering have been very successful, but they only address about 1/3 of the problem, and I'll expand a bit on that. And we believe that ARTPEP can fill in and address a big part of the 2/3 residual risk. So about the context, you can learn a lot by watching, and sometimes you have to watch for a long time. Already in 1951, 2 years before I was born, Bar, Eder, and Russ, and I actually worked with Eder for a while. He was from the Midwest and worked at NIH, showed that patients with low HDL cholesterol run higher risk of getting myocardial infarcts. 77 in the Framingham Heart Study, they showed that both the LDL cholesterol concentration makes a difference. Higher HDL, higher risk of getting myocardial infarct. But even when LDL is low, for example, by statin treatment, the HDL cholesterol concentration makes a difference for risk of getting myocardial infarct. So this has coined the idea that LDL is the bad guy and HDL is the good guy, sweeping away atherosclerosis. How efficient are really statins? Well, statins really address 1/3 of the problem, and one key reason is that it takes a while for the statins to kick in. This is a study in acute coronary syndrome patients, and the statins take about 4 weeks to kick in, and during those 4 weeks, 2/3 of the events already have occurred. So in the treatment of secondary prevention, it is very important to have a quick onset of action by your drug, and I'll show you that our peptide has a quick onset of action. In this particular study that I showed, which was an LDL lowering study, it still showed that the concentration of HDL and ApoA-I, which is the main protein in HDL, were predictors of a new event. So many studies have been done with statins. They've been large. They've used max doses. They've been controlled and very well performed, and still 2/3 of the problem is still there, and that is what we're working with. What about the target? If you do not where you're going, you'll end up somewhere else. So the target is very important. The target of HDL, and really the active part of HDL, pre-beta HDL, is the ApoA-I transporter. People with the ApoA-I transporter deficiency, they have what's called Tanger's disease. They get premature cardiovascular disease and improved ApoA functionality. By HDL and our peptide will regress atherosclerosis and prevent cardiovascular disease events. This, on top, you can see a artery. You can see the core plaque, which is full of macrophages and lipid, and you have the typical rupture at the shoulder of the plaque. Now, in that shoulder, you have an accumulation of macrophages, which are cholesterol-filled, thereby they have changed phenotype. Cholesterol is toxic, so in order to defend themselves, the macrophages, as they acquire cholesterol, express the ABCA1 transporter, which is the rate-limiting transporter for removing cholesterol. And the concentration of this transporter is also dependent on whether this transporter has a ligand, for example, a peptide or pre-beta HDL. This is kind of how it works. So the nascent forms of HDL or pure ApoA-I docks with a ABCA1 transporter and picks up free cholesterol from the cell membrane. And in this process, the cholesterol esters, which constitute the cholesterol pool, is being diminished, and thereby the macrophage change phenotype. So the concept of pre-beta HDL infusions have been tested 3 times, first in a study performed by a company I co-founded, ApoA-I Milano, and the Artery Therapeutics company, which was sold to Pfizer, and it showed that already in 6 weeks in humans, you could remove substantial amounts of atherosclerosis. You could actually remove 2 years worth of atherosclerosis in 6 weeks. And 2 other companies used the same protocol using another technology, and here also wild-type ApoA-I, and all 3 consistently found that in very short order, in very short time, you can remove substantial amounts of atherosclerosis using pre-beta HDL. A problem with all these 3 technologies is that they are recombinant ApoA-I technologies where you have complex the protein with phospholipid, so we have lots of stability cost and potentially safety problems. So we have, and John Balicki in particular, has developed RPEP2, and it is potent in removing free cholesterol from macrophages, in this case J774 macrophages. It is as potent or more potent. You'll see EC50 values later on. It stabilizes the ABCA1 transporter. The signal here with RPEP is about twice as high as that one without RPEP. When you look at an EC50 type of curve, you see the very potent efflux, and when you calculate the EC50, it is 0.8 micrograms per milliliter, which is about 5 times more potent than ApoA-I, and ApoA-I is nature's most potent ABCA1 ligand. So this is a fantastic result. This peptide also has another very interesting feature, namely when you incubate human plasma with a peptide and then you illustrate this on a gel. This is a 2-dimensional gel, so migration according to charge and according to size. When you look at it after incubation, you have an increase of ApoA-I signal in the so-called pre-beta area, which is the important species. When you detect this with an antibody to the peptide, you see that the peptide only distributes in this alpha migration area, not in the pre-beta. So what the peptide is doing is that it's binding to the alpha particles and cause a reconfirmation and conversion of ApoA-I from alpha particles to pre-beta, to the active particles. So the peptide has effect in itself, and it is supercharging existing HDL. And that can also be illustrated when you spike peptide into serum, and then you get an additional ABCA1 efflux. Now, whether it's truly caused by the peptide or the pre-beta generated by the peptide, is very hard to figure out. Now, when it comes to animal studies, one way of assessing efficacy is to inject macrophage foam cells which have been filled with triturated cholesterol intraperitoneally, and then follow the triturated signal to serum through the liver and to feces. And what we see here is a dose response increase in the fecal excretion all the way through the kidney and out to feces. And that at very low doses, 10 micrograms per kilo, which is a low dose in an atherosclerotic ApoE knockout mice. So when we look at atherosclerosis, this peptide is very efficient. It reduces, in this case, 32% of atherosclerosis. So this is a engineered mice, ApoA-I knockout mice, which receives Western diet for 14 weeks and becomes atherosclerotic. And then we treat for 6 weeks with peptide or with placebo. And you see the difference here for whole aorta, the thoracic and abdominal. We have another peptide which is equally efficient, but we have continued to develop ARTPEP, which is the green bar here, for various reasons. So here is a second study with a lower dose, 10 milligrams per kilo given every other day. This peptide has a long half-life, so it's possible to give it every other day. And you see also at this dose a significant decrease in atherosclerosis. So when we compare these two studies, the 6-week study and the second study, which was a 10-week study at doses 25 milligrams per kilo IP and 10 milligrams IP, which corresponds to 10 and 3 milligrams per kilo IV, you can see that it compares favorably to the A1 Milano complex, where 40 milligrams per kilo IV gave a similar type of atherosclerosis reduction. So we are very upbeat about this because the peptide is given as a free peptide, while as this is a complicated product with a recombinant 243 amino acid protein complex with phospholipid. So ARTPEP is druggable, done by simple FMOC peptide synthesis. It's administered as free peptide. The concentration can be 75 milligrams per milliliter or higher, and that is because in high concentration it distributes as an oligomer, and at low concentrations as at a monomer. It can be given subcutaneously because it has a 33% bioavailability. It binds to HDL and assumes day-long half-life. It is efficient in reducing athero from 10 milligrams per kilo. And in tox studies, we can see that at 500 milligrams per kilo in rats, multiple dosing, we see no tox in serum or in tissues. So in summary, it's a potent, safe, and convenient peptide with favorable drug properties. EC50 is low, 5 times better than nature's best peptide. It functions in vivo, and it's non-toxic. We have a good safety margin. It can be administered in a very convenient way, once weekly and subcutaneously, and it's ready for IND-enabling studies. Clinical projections is to use this peptide in secondary prevention to patients that have had an event like a myocardial infarct or acute coronary syndrome or stroke. It can also obviate the need for coronary invasive procedures like stenting and PTCA, coronary bypass. We believe the regimen will be weekly injections for 6 to 16 weeks. It has patent protection for 20 years. The market size is plus 10 billion per year. And there is a need for tons of material, of course, if this becomes real. So what about the future? It's tough to make predictions, especially about the future, but it is the largest therapy area. It's a big unmet medical need. It's a validated target, and we have good mechanism of action understanding. It's potent, selective, and a druggable peptide, and there is a scarcity of other products, and we are a good team, and we have two team members here. We have 3, including myself, but John Bailicki, and we also have Walid Dano, who's helping us since he retired from Roche. And besides that, we have Prenman Shah, who's been doing a lot of animal studies with A1 Milano and so forth. And this guy is the former head of the Nobel Prize awarding committee for 15 years. So he knows a lot of peptide chemists, more than 700 peer-reviewed papers. And I'm ready for questions.
Thank you, Chris. We do have time for questions.
So if you need 10 milligrams per kilo, that translates 700 milligrams per patient, and you have solubility 70 mg per ml, so that's 10 milliliter injection. Is that correct? Is it a problem in the clinic?
Well, we know a fair amount since 3 pre-beta prototype drugs have been tested, so we know a bit about how to allometrically scale the efficacy side of this equation. We believe that the required dose will be about 100 milligrams per patient per week.
And what's the pharmacokinetics? Does it persist for a week, the peptide?
It has a half-life, estimated half-life in humans. We haven't done human studies, but based from mice and rats and other species, of 3.5 days in man.
It wasn't clear to me, is the Experience Pfizer peptide still being pursued commercially, or is that just too complex?
That was a full recombinant protein, 243 amino acids, ApoA-I protein. It was sold from Pfizer to the medicines company, and the medicines company had to go back and redo the production vehicle, basically remove the endotoxin generating parts in the E. coli, and it's now being pursued again because it had such a powerful effect. And whatever really works and affects disease will be pursued because people will pay for it.
If there are no
Thank you.
further questions, let's thank Chris again.